Our next speaker is Peter Shaw with Merck, who will be speaking from the pharmaceutical perspective.
Thank you. Thanks to the organisers for inviting me. So what I'm going to do today is I'm going to talk about some scenarios for developing personalised medicines, give you some examples, and really talk about, the evolving field to deliver personalised medicine and some of the issues that we see as we move forward. Now, there are two scenarios, I think, that have really occurred over the last few years. And the first scenario that I'm depicting here is really an optimal situation. And the optimal situation is when you understand the biology right at the start of a development program and you have a target that you know is either expressed in a fraction of a population or is active in a population, and you develop a drug to that. When you have that situation, things are pretty much aligned in where you can actually develop a test along with the drug to identify people who you want to bring into your clinical trials, and as well get the test and the drug approved at the same time. And why that's critical really is when you have a test and the drug approved at the same time, the uptake into and use of the drug is really works well. And there are several examples of this that have occurred over the last few years. Classic one, the first one is Herceptin, where you have an amplification of the HER2 gene, which is measured either by an IHC assay or a FISH assay, and then some more current examples, crizotinib and vemurafenib. Each of these have molecular markers that identify a subset of a cancer population. And again, what you can see here as well is that many of these examples are actually in the cancer population. This might not be surprising. Cancer is a genetic disease, and in fact, we're going to see a lot more of this as we go forward because we now have the technologies to identify these mutations and associate them with some of the phenotypes. So we can expect to see a lot more of this as we go forward, but there's only a few real examples over the last few years. Now, this is the situation that typically happens, and this is more often what we're looking at. And what we see here is where we don't have all of the information early on in the drug development paradigm. We just don't have enough evidence to associate a molecular marker with the drug response. And in fact, what happens typically is we get this data later on, either in phase 3 studies when we unblind them, right at the end when we're filing, or later on when the drug's actually approved, where we see other studies that have been done which point to molecular markers that might actually be useful for identifying subpopulations. And this is quite challenging because what you have here is you have emergent data that you will need to validate or refute to make sure that it is scientifically valid. And dependent on when that comes out in the time frame, it's harder to change clinical practice. So the further away you are from the approval of the drug and the data coming out, physicians are already used to using the drug, they feel familiar with it, and they don't really want to use the genetic information. So there's lots of issues with this, and I think we're going to see a lot more of this as well in the next few years. And I'll point to some of the factors that are going to drive that later on in the talk. So here's some examples of data that's come out after a drug has been approved. Now, this is a really good example. It was a Schering-Plough example. And what they did is they did a GWAS on some phase 3 studies, and they found this genetic marker in the IL-28B gene, and there's several other markers in linkage disequilibrium, which actually, if you think about them, they point to a twofold increase in the response rate to PEG interferon therapy. So patients with the CC genotype have a twofold better chance of responding to PEG interferon. This was a fantastic discovery. And in fact, there is tests available for this. And for patients who are being treated with PEG interferon, this actually is used by physicians to actually persuade patients to go on to this therapy, because it's a tough therapy to actually endure. It's about a year, and it's really hard on the patients. However, now we actually have different therapies that have been approved. So over the last year, there was a third agent that is added to this regimen, another direct-acting agent a protease inhibitor, telaprevir and boceprevir. And in fact, what we see is that when you add that agent to this PEG interferon ribavirin therapy, those patients in the TT and the TC genotypes actually respond a lot better now when you add that protease inhibitor. So treatment paradigms have now superseded this really nice genetic finding to a certain extent. Here's another example, and this is warfarin, and this was talked about earlier on. And this is, one of the most widely prescribed anticoagulants. And what's been clear, at least in my mind over the last few years, is that this drug, it's really important to get the dose right when you start up. And physicians use demographic and clinical factors to actually pick the dose, and they might have to titrate the dose, in fact, to get it right. And what some of these investigators did, as evidence became available, is they started to look at the impact of these two genes, 2C9, which is involved in the metabolism of warfarin, and actually VCOR-C1, which is the target for warfarin. And in fact, when they took genetic information and added it to the clinical parameters that are used for dosing, what they show here in the chart is that, in fact, the genetic information is quite useful, in fact, at picking the dose for those people who require lower doses of warfarin and for those who require higher doses of warfarin. So this information is quite important. In fact, from my standpoint, if I wanted to go on to warfarin, I think I'd want this information. But how many people do you think have that genetic test done, or if that algorithm is used? I'm not sure what the number is, but I bet it's not everywhere. Here's another example in cardiovascular. And this is, I'll do a few slides on this one, because I think this is quite interesting too. And this is to do with the drug Plavix, clopidogrel. In fact, clopidogrel is a prodrug. So if you look at the chart here on the top, it's absorbed. Most of it's actually excreted, and a small fraction of it is activated in the liver by a gene called 2C19 for the most part. The active metabolite does its business by binding to the P2Y12 receptor here, which prevents platelet aggregation. So that's the basic fundamental mechanism for this drug. This is some of the data that came out by Schuldiner. And in fact, here's a Manhattan plot, and what they show here quite nicely is the locus 2C19 with some other CYPs, but primarily 2C19, is associated with platelet aggregation. And in fact, there's some more data here that shows you that if you carry a non-functional variant of the 2C19 gene, you have more aggregation, i.e. the drug is less effective if you have those non-functional mutations. Now, Eli Lilly and Daiichi Sankyo developed another compound, I never get this right, thienopyridine, similar to clopidogrel, and they did a very large study here to demonstrate the effectiveness, the Triton-Timmy study, in 13,608 patients. And this data here, which was described in the New England Journal of Medicine, quite nicely shows that for efficacy, prasugrel is better. There are less cardiovascular events here than for clopidogrel. But on the safety events they measured, actually prasugrel did a little worse than clopidogrel. This data was really nice data, and they then did a sub-study where they looked at the genetic variants, knowing that clopidogrel is metabolized by 2C19. And what they showed here, quite convincingly, is that if you carry a non-functional allele of 2C19, there's a higher risk of an event. And then over here in healthy volunteers, they also showed that if you have non-functional 2C19, you produce less active metabolite. These are healthy volunteers. And again, there's a pharmacodynamic assay here which shows that the platelet aggregation is worse if you have the non-functional allele. Now, this data, with other data sets that was in the literature, was then used to make labels. And these are some of the label excerpts that I've just pulled out from the prasugrel label. And this is interesting because this is the first time the FDA actually, because they had all the data from the Triton-Timmy study, actually mentioned another drug. And so here you can read it. Both prasugrel and clopidogrel are prodrugs that must be metabolized to their active moieties. The pharmacokinetics of prasugrel's active metabolite are not known to be affected by genetic variations. And then the pharmacokinetics of clopidogrel's are affected by 2C19 genotype. Okay, so that's the prasugrel label that mentions clopidogrel. That's it, I got it here. And then here is the clopidogrel label. And again, I pulled out some interesting facts from the clopidogrel label. And again, you know, the effectiveness of Plavix depends on the activation by the P450 system, principally 2C19. Poor metabolizers treated with Plavix at recommended doses exhibit higher cardiovascular event rates. Everything that I've told you, which was produced in those New England Journal of Medicine. Tests are available to identify a patient's 2C19 genotype and can be used to determine a therapeutic strategy and then consider alternative treatments for patients identified as poor metabolizers. So that paints a clear picture, right, of what we've got to do. Here's the article that came out in the Wall Street Journal. And these are quotes from, I think, leading academics and clinicians. And you can see here that, you know, not everybody's thinking about this in the same way. In fact, you know, many doctors are scrambling to figure out which heart patients should continue to take the top-selling drug, Plavix. There's more than enough data now to guide an individual patient on Plavix. I like this one. At Duke University, patients can get the genetic test if they ask for it. Now, how many people do you think are going to go in and ask for this test? No, so, you know, it's kind of, I think it's sad in some senses where we've got really good science going on, but we have a communication problem, I think, in taking that science and thinking about it and then putting it into clinical practice. So the one message I have really is we have an education issue that we need to deal with. When we're doing all this good science, whether it's for simple genetic markers or for complex markers, we need to translate this into simple messages that can be used to improve our health. Now, from a pharmaceutical perspective, we get this, and we're under enormous pressure, really, not just from my boss, but from practitioners, payers, and regulators to really demonstrate the value of our drugs. And we know that the molecular identification of subpopulations is probably one way, not the only way, but one way in which we can do this. And so we are very much interested in doing this. And why would we do it? Well, the first thing is we want to identify the patients with the greatest unmet medical need. And if there are molecular markers that can do that for us, those are the patients we want to be treating. We don't need to be treating patients who are effectively managed, who have good drugs that are working. If we can use these molecular markers to identify patients, we can enroll those people into our clinical trials because it's a hard job to find a signal of efficacy, especially when you're comparing to the standard of care. So we want to try and see that signal to get our drugs approved. And this is one way we can do it. It may help us to shorten our trials, especially if there are markers that really identify patients who will do well on drugs. As well, as we get more information, as we go on to phase 3 and in the post-marketing situation, it may help us optimize the product. So we refine the patient populations that should get this drug. And if we can do all these three things, we believe that we can increase compliance and cost effectiveness of our drugs, and potentially differentiate from our competitors. So what are the concerns and barriers? And you know, you hear this from different people. There is a substantial investment required because fundamentally what we need to do is understand how these biomarkers are impacting drug response. And we've heard a lot of talks today. There are very few single genes that tell you how a drug's going to work. In fact, you know, some of the ones I mentioned only account for 10% of the variability of drug response. And that's pretty big for a single marker. So we do need to do some more complex biology and bring that together. And it's going to require substantive investment. The other issue is that people say that it's, you know, if you want to identify a patient with a molecular marker, then it's going to be more complex screening. And it is. So you have to take the population and find out who is actually expressing this marker, who has this genetic variant, and that takes increasing time, especially if the assay is really complex and it requires a lot of analysis. The other thing is that when you need a test, and you actually have to have a parallel development program. So you have your drug development program, and you have to develop the IVD, or the test, and you have to integrate these. And that is actually much more complex. It's not so easy. The timing, you have to get everything right, the data set's right, the validation of the test, all of this have to move along in a parallel fashion so you can submit the data to the regulators at the same time. There are concerns over limiting market size. I don't believe that because, from my standpoint, if you can identify a population who needs a drug, then you have value. And, so limiting the market size doesn't make sense to me. It's all about developing drugs that have value to patients who need them. The other issue that comes up are the cost of the test. And I think this could be an issue. You've got a variety of costs. Some tests cost peanuts, $1. Some tests cost $5,000. And when you add that to the drug cost as well, it could be a barrier. However, I think that the drug industry and the diagnostic industry can actually sort out decent business models in the end. It's all about the value to the patient. So, targeted medicines aren't for every drug either. And what's critical from the pharma perspective is trying to sort through this information as early as we can so that we do co-develop tests with the right drugs. So what are some of the enablers and drivers that are out in the field? And, there are at least 6. I've depicted them there that are all coming together to hopefully really enable personalized medicines. And we can just look at a couple of them in detail. The first one is the Genomic Technologies, and we had a nice talk by Eric this morning, Eric Schatt. And what we do know is that the cost to actually develop raw genomic information is decreased incredibly. And so we are in a position now to really generate this information rapidly and cheaply. What does cost money, actually, is putting it all together. So when people talk about the $1,000 genome, that's really just the wet biology and the chips and everything to actually generate that information. We've got to make sense of it. You've got to put it all together. And that is going to cost some money at the moment. And we have to think about better ways to do that faster and at less cost. I think it's really interesting, though, because I think having this information is going to help us understand the disease biology, the contribution of rare variants to complex biology. The next thing is, what are the regulators doing? And in fact, they are driving personalized medicine. And this is a slide that I put together. And, I just kind of put together some examples of where, you have a drug and you have a genetic test. And you can see that there are a lot of examples in oncology, but there are some in cardiovascular too, infectious diseases. So it's happening everywhere. What I think is important is this question of utility. So these tests, whatever they are, single genes, complex transcriptional biomarker sets, they have to provide a simple decision for a physician to use the drug. So you need algorithms that are taking complex biology and sorting it down into probably, what dose do I need to use for this patient? Should I give this drug? Should I give drug B? And that's where we're going to have to think about algorithms that reduce complex biology into simple decision-making tools. Now, I think the future's kind of bright in some sense. In drug development, we have our paradigm of phase 1, 2, 3, and life cycle management. And our clinical studies that get our drugs approved are only in a very small fraction of people compared to the exposure when they actually get approved. And in fact, what you see happening now is that medical centers, and these are just a few examples, Geisinger, Kaiser, Vanderbilt, what they're doing is they're going into there and developing electronic medical records where they can pull out phenotypes of patients who are being treated at their hospital center. And so they have sophisticated tools now where they can actually pull out the phenotypes of interest, and then link it now, because the cost of doing genetics has gone down. And they can do these pharmacogenetic studies quite rapidly now, once a drug's been approved. And I think this is where we're going to see, as well, a lot of data coming out in the next 5 to 10 years, because they have, not only are they doing it at their own centers, but they're joining larger networks. And here is where you really have that statistical power, where you're looking in thousands and thousands of people when these groups get together and collaborate. So the consequence, you're going to get rapid generation of PGT data sets post-approval, outside randomized clinical trials. It's going to be good data. And then these centers are actually going to develop their own algorithms as well for teaching patients. When they find out that genetic markers are relevant to response, they're going to start incorporating them. It makes sense. Here's another example. And this is the International Serious Adverse Events Consortium. It's mainly sponsored by pharmaceutical companies, but there are many members. So people from Kaiser are present in the consortium. And really what I wanted to show you here is just another approach to tackling this problem. These serious events, they occur at such a low frequency, we rarely detect them in our clinical trials. The drug's out on the market, and suddenly we start seeing some of these events because of the exposure in the broader population. And it's quite difficult then to go and get a genetic sample from these people who are in disparate places. But what we did through this consortium and under Arthur Holden's real guidance here is he was able to go to different centers who were collecting samples from individual patients all over the world. In the UK, for example, and other places in Europe, China, et cetera, and pull together all of these cases. And again, that's where you start to have the power to do these genetic studies and understand, what other genetic factors, are determining the susceptibility drug-induced liver injury to not just one drug, but to multiple drugs. What's the contribution of ethnic diversity? And I heard, in the previous talk about the case control design. That's critical because this ethnic variability, can really be important in driving the conclusions that you make. Other people are interested in using pharmacogenetics, other stakeholders in the healthcare industry. And this is just one example. So Medco, they have their own research group that's dedicated to understanding the genetic contribution to drug response. In fact, they've got a lot of studies ongoing at the moment that try to identify, and one example is with Plavix and clopidogrel. They're doing a study themselves to understand the contribution of 2C19 to response. And, their business model is that, if there is a genetic subpopulation who do well on a drug, so, for example, 80% of the people receiving clopidogrel, and they can identify them with a genetic marker, they should stay on that drug. Makes sense. And they can pass those cost savings on to their customers. And, it's interesting because, if that works as well, it's a challenge for pharma because we may have a pivotal phase 3 running, which takes 3 to 5 years, and the bar may change. So if we're doing a comparator with Plavix, just for example, the bar of what is the efficacy rate would change if you have a subpopulation who do really well and you take out the people who don't do so well. So, that poses some challenges as well. Okay. So just kind of pulling this all together, we have to think about this and think about the evolving field, in a 2 to 5 year perspective and even longer. And this is just one way to think about it. And the reason we have to think about it like this is it takes 10 years to develop a drug. So we have to kind of think, well, what's the field going to look like in 10 years' time? Because that's the kind of information I'm going to need and start thinking about developing today. And, I kind of gone through this, but, we think sequencing technology is the technology of choice, or will be. The $1,000 genome is approaching. We still got some dollars to actually sort out that information. We think that rare variants are going to be catalogued, and the Broad Institute has gone a long way to doing that. They've sequenced thousands of people already and have catalogued rare variants. And you're going to see a chip come out fairly soon, I think, from Illumina or one of these companies that have all these rare variants on it. So this is already happening. EMRs, they've already been implemented at many healthcare centers. They're pulling phenotypes. They're doing really nice pharmacogenetic experiments. They're validating results that we've seen. They're finding new discoveries. The regulators are pushing hard on personalized medicine. It makes sense. Right drug, right patient, right time, right dose. So they're encouraging us to put that information into the labels. You're seeing more of it. And there will be, requirements to actually generate this information going forward. Then you have the providers and payers also, thinking about how to use this information for their own decision making. You have the body in the UK, NICE, who scrutinizes the value of drugs. So you can just think about this being extended into ADME variants and genetic populations who will and won't respond. I suspect that will be coming. Personalized medicine companies, new companies thinking about delivering this information to the consumers. It's hard to know what role they'll play going forward, but they're certainly collaborating with academic institutes, thinking about ways to deliver information that's useful for decision making. The farmers, we're definitely using the technologies. We've gone past oncology now. We're looking at other therapeutic areas, and so you could expect to see more of this in the future. So what are the take-home messages? For me, personalized medicine's arrived. I think there are some challenges going forward. Other stakeholders have the tools to develop pharmacogenetic data. So we have to think about all this data coming in and how do we use it and focus it to really help treat patients in the right way. So there's going to be studies coming from academic centers, from these EMRs, and we need to consolidate that information in a way that's useful to patients. This is going to require, integration and critical thinking across different functional areas, not only in pharma, but with other external stakeholders who are developing this information too. And I think, the future for generating robust data is here. So thank you. I should, acknowledgements. I want to thank the people.
Peter, I think we don't have any time, but we will take the time for 2 quick questions.
Thank you. Albert Donegan, has your company or any other pharmaceutical companies you know of considered the potential for doing exactly this type of approach through drug development to drug marketing, but with a much less expensive technique of breath analysis by GC? The variability is a lot smaller, approximately 4,000 compounds identified so far at the PPT level, and humans only share 27 of them. So there's a lot of noise of exposure and disease to look at, and you could in the same fashion be breath profiling people before they start phase 1 and 2 trials, identifying at the end of those trials which a priori breath prints fit good responders and bad responders, and it's a lot easier to give a GP in their office a breath analyzer than to give them all the tools for this genomic analysis, and they can run the breath in 20 minutes.
Yeah. So, I don't know the answer to your question if we've done it or if any other company's done it. But, as a platform, I think it sounds like it has potential. And I think, I want to follow up on the fact that the platforms that we use to make the discoveries, the genomic platforms, the GWASs, and things like that, today we have to reduce those into simple decision-making tools. So they might be good platforms for discovery. They might not actually ultimately be the test that's in the office. It might kind of reduce to something else that's simpler.
I think their huge weakness, though, all these genetic tests we've learned today are not giving us necessarily the phenotypic outcome, the metabolic outcome of their genetic starting blocks. Whereas if we look at metabolomics in breath, we've got the outcome, which is what we're trying to parse in terms of who's a good responder and a bad responder. And so instead of finding genes that are associated with a few % of a disease, we're finding profiles that are already sensitive and specific enough to replace some traditional screening tests like breath prints instead of mammograms.
Yeah, and I don't disagree with you. I actually, I think that I talked about the genetic tools because it's easier to kind of use that as a paradigm, and there's good examples out there. And I think any platform that can do that, reduce complexity, or provide additional information to genetics, because we know that a lot of these markers, as I said, only contribute to maybe 10% or less of the variability that we're seeing. So we know that there may be other better ways to do this. But
okay, thank you.
Well, I think, thank you very much, Peter.